for those of you who don't know me, I'm Rachel Heilman. I'm the Director of Research for the Med13L Foundation. Um, and I'll be presenting last today. Today we're partnering with Simons Searchlight on an update for their data as well as EEG updates, which were gathered at the 2022 Med13L Simons Searchlight Collaboration Family Conference, which predates me. So I'm at least in conjunction with Med13L. So we're really excited to have that, and then lucky to have researcher Randy Strick join us as well from Rowan University. With that, Cora, I will let you go ahead and take it away with our Simons Searchlight family data updates.
Great. I am sharing my slides here and starting my little timer because I try to keep good track of where my time is at. So for those of you who haven't met me, my name is Cora Taylor. I'm one of the principal investigators on the Simons Searchlight project. It's a multi-site study. So there's us here at Geisinger. If you're participating in your portal or you're first enrolling, you're likely interacting with somebody on our team here at Geisinger. And then also Dr. Wendy Chung at Boston Children's Hospital is also another principal investigator on the project. So happy to hopefully be talking to some of the families that I maybe met back in 2022 when I had the privilege of being there and meeting your families. And then just sharing a little bit of updates from what's been going on since that time. All right, I should check before I keep going that everybody can see my screen okay. And I can't see faces, so a verbal yes would be great. Oh,
Yes.
perfect. Okay. For those of you who aren't familiar with Simons Searchlight, we're an international research program to better understand a range of rare genetic conditions that cause neurodevelopmental disorders, of which Med13L just happens to be one of them. So we do try to bring in families from all over the world. It's very important in rare genetic disease research to try to engage individuals across the United States, but also across the world, because many of the conditions, one of which Med13L falls into this category, is exceptionally rare or potentially underdiagnosed. So we want to try to get as many families involved as possible. As you can probably imagine, in order to make that possible, it takes a bit of a village. So there are individuals from multiple organizations that participate. So the Simons Foundation absolutely contributes, provides funding for our project, and they also oversee some of the scientific aspects of our project. Boston Children's Hospital also heavily involved. Us here at Geisinger in central Pennsylvania. And then we also work very closely with a agency called Tempest, which does all of the online platform work for us. So it takes a lot of people on our team to make sure that this all goes off as best as we can. So overall, if you're not familiar with Simons Searchlight, what are the overall goals? So we have kind of a numerous goal. So one is that we want to better understand what we call the natural history of the study, both from a medical standpoint. So what medical conditions do we see that are more common in individuals with Med13L, but then also behaviorally. So are there increased psychiatric or developmental conditions that are associated with Med13L? And what rates do we see those conditions based on what we're hearing from families? We also collect blood samples for families. That is an optional part of our participation, but I hope I can convince you today, if you haven't given a blood sample, why that's a really important part of participation and how it's valuable to researchers and moving things like clinical trials or treatments forward. When you do submit your data to Simons Searchlight, we do try to synthesize that information back to families and help you understand how your individual child with Med13L might fit into the broader picture of Med13L and what specific unique challenges or strengths your child might have. We do also feel very strongly about the concept of freely sharing data. So anytime you submit your data to Simon Searchlight, we share it back currently on a quarterly basis to research other researchers who are external to Simon Searchlight. So that previous slide where I showed all of our faces, we're not the only ones who would necessarily have access to that data. Other researchers that you're hearing from today could log on to our website and access that data. If they have trouble doing that, they can absolutely reach out to our team and help understand that process better. And then also just in general, connecting researchers around the world and promoting better understanding. So I mentioned very briefly earlier that the Simons Foundation is our funder. The Simons Foundation offers a number of different streams of funding for a number of different initiatives, of which Safari or the Simons Foundation Autism Research Initiative is just one of them. There are multiple projects that fall under that umbrella. There's the Spark cohort, which some of you might be participating in. That's for anyone who has an autism diagnosis, regardless of whether or not they may also have a genetic condition. Simon Searchlight, while autism is in the name of our initiative, Simon Searchlight does not focus solely on autism. And that's a point I want to highlight for your entire community is that having met some of your families, I know that some of you have an autism diagnosis and some of you may not, but having Med13L in and of itself makes you eligible to participate. So even if your child does not meet clear clinical criteria for autism, or if they don't appear to have autism at all, they are absolutely eligible to participate in Simon Searchlight. There are a few other resources that I'm not going to get into in as much detail today just due to time. So here are the overall conditions we study. So currently we have nearly 200, and there are other languages that people can participate in as well, other than English, including Spanish, French, and Dutch. We're working on Italian and Portuguese as well. Just taking a quick look at the kind of information that we collect. So after you enroll, we collect a bunch of information at what we call baseline. That just means it's your first interaction with us. The first thing that we collect, and it's sometimes the place where people get hung up, and we would like to try to emphasize as much as we can that we will help as much as we can at this point, which is that we need clinical genetic lab results. The reason for this is that we want to make sure that everybody that we're enrolling in our study truly does have a variant in Med13L that may be clinically causing their symptoms. So sometimes we'll get a result back and it says that genetic result is benign, so we know it's not causative. And we want to make sure that people are coming in the right way and that everybody in the Med13L community is in the right community. They're where they should be. In terms of interview and online surveys, we do collect medical histories. For those of you who've been participating with us for a long time, you might know that those were traditionally handled over the phone, so you get an annual phone call. We're working on transitioning that towards being entirely online, so you don't have to try to schedule a phone call. We do that both at your first visit and then also annually. So if there are additional medical concerns that have popped up, we can have time to talk to you about those. And then I'll just let you briefly look at these other ones. So some of the online surveys, we ask about sleep, we ask about seizures, we ask about day-to-day living skills, behavioral skills, and then also symptoms related to autism and then overall quality of life. Again, like I mentioned before, new survey, the medical history survey is fully online. If you were to complete this fully online, and our team is currently reviewing them as well. So if we had questions about some of the things that you had mentioned, something didn't make sense, or we didn't understand how you answered the survey, we would call you and follow up with you. If you are not a participant, this is essentially what your dashboard looks like. So if you log on to your account and see what research steps you have to participate in, you have kind of this look at the top that shows you what you need to do, what you need to do next, and what tasks you need to complete, not just for yourself, but potentially for other members of your family. So if you have enrolled your child and you might need to complete surveys, this will all show up in your overall what we call a dashboard. And then of note, we do provide Amazon gift cards after survey completion for everyone. Here are a few things that are coming up. So this is not things that are we are expecting to have done by the end of the year, but things that we're hoping to have done within approximately 2025. The first one, and I think one that is of particular significance to the Med13L group, is this Observed Reporter Communication Assessment, or the ORCA assessment. It's specifically designed for individuals who maybe have a lot more trouble communicating verbally. So some of what we've learned from our families is that some of the assessments that we have been using to collect information about language are not capturing those kids who are having trouble communicating. We know from working with our Med13L families that we have some individuals who are nonverbal. We have some individuals who are using a communication device or might be trying to use sign language. And there are some individuals who I met personally in 2022 who are what I would call a multimodal communicator. So they are using a combination of verbal speech and using a device. So our hope with the ORCA is that we're going to be able to get better information on those kids who are not falling cleanly within absolutely verbal speech. They're talking, but what other ways are they communicating? So if we were to institute different kinds of treatments or interventions, that we can understand how those might impact their scores on something like the ORCA. We're also working on better understanding behavioral problems that you're seeing. So we want to have a behavior problems inventory implemented within the next year, an overall developmental milestone update. So age at walking, age at talking. So if you have a very young child who's enrolled in Simons Searchlight, they may not be talking at the first time you complete your history, but we want to give these brief updates so that you can say, oh, my child is talking now, or they are walking now. Uh, Also getting a better understanding of overall disability and working on ways to collect EEG records and data over time. That is likely to be a more long-term project than short-term. This is just a sample of the kind of information that we're providing back to families. So on a quarterly basis, although again, we are working on trying to make this a little bit more ongoing. Right now, this is just an example of the kind of information that we're giving back. So it's kind of just a couple pages just about the types of issues that we see in families with Med13L. We also participate in what we call Research Match. So this is a way that researchers who are external to Simons Searchlight can contact individuals with Med13L and ask them if they want to enroll in their studies. We have a variety of different ways that researchers can reach out to our families. If you hear from a researcher and they say, I really want to access this group of individuals with Med13L who've already participated in Searchlight, how can I reach out to them? I would encourage you to get in touch with our team, and we can help facilitate the best avenue for that researcher to get in touch with us. I'm going to briefly revisit blood samples. So for those of you who were at the meeting in 2022, you may have participated in a blood sample that we collected in person. If you are not attending a meeting, or if we are only having virtual meetings for a while, you can provide a blood sample from your home. We partner with Quest Laboratories. We mail Quest Laboratory kits to your home. You make an appointment, and then they just put them back in the mail. So that is an option. There are a number of different cell types that we collect from the blood samples. The one that I want to focus on briefly is what we call induced pluripotent stem cells, or what we call iPSCs. These are really powerful tools for research. The reason is that we take a blood sample from a participant, and then by going through some reformatting, we're able to turn those into a researcher is able to turn those into any kind of tissue cell. So for example, given that we know that Med13L impacts the way the brains develop, we might have researchers who are very interested in studying neurons in individuals with Med13L. So by drawing a blood sample, we can reprogram them, turn them into neurons, and then it facilitates some basic research to better understand how Med13L is impacting the brain. We do have a repository. It is a partnership with the New York Stem Cell Foundation, where we are creating the iPSCs ourselves that are available to researchers. Not every variant is necessarily represented here. We've been trying to pick and choose which ones are going to kind of become the iPSCs because there is just a limited capacity to do that. So here's what is available to researchers. So right now, there are a number of different institutions that are using all of our samples, including the data sets, the biospecimens, as well as neuroimaging. And Med13L is represented across both phenotypic data sets and biospecimen samples. I'm going to go ahead and just quickly pass this slide, but again, like I mentioned before, if any researcher on the call or any researcher that you know is interested in accessing our data, the website is base.safari.org. But if you're having trouble, please get in touch with our team because we're more than happy to help in that regard. Okay, so with my last bit of time here, I would like to switch gears and focus specifically on the Med13L registry. So not the overall project, but what do we specifically know about Med13L and who's participating based on the data that we have in Simons Searchlight right now? So here's overall. So at in the different steps is what individuals are doing. And in those little blue boxes are the number of individuals who've made it through the different steps. So you'll see that we have 164 individuals who've signed up online and said that they have a family member who's impacted by Med13L. Of those, 129 have provided a genetic lab report to show definitively that they, in fact, do have Med13L, a pathogenic change or a variant of uncertain significance, and that they are eligible to participate in additional steps. Of those, we have 87 who've done the medical history background, keeping in mind that this used to be done on the phone, which is why we probably have more people who've actually filled out surveys versus those who've done the medical history, because it's a little bit of a barrier to do the phone and scheduling versus just being able to do it online. So hopefully we'll see those numbers increase with the new online survey. We have 31 individuals who have provided a blood sample as well. So, uh, just of note, we have 39 more individuals that are participating in Simons Searchlight this year compared to the data that we presented to you in 2022. So your community is continuing to grow. This just shows an overall just the length of the gene and where the different mutations are located. In addition, and just as a note at the bottom, we do allow individuals who have variants of uncertain significance to participate in Searchlight, but for the purposes of our presentation, we do not include them in the data that we're showing. That's just because we're not sure if that Med13L variant is causative of their symptoms or not. Here's where you guys are located. So you are one of our communities that is all over the world. Perhaps not surprisingly, most of you are located in the United States, but we do have individuals everywhere. And hopefully, as our languages expand, we'll be able to include more individuals from more areas of the world. If we zoom in a little bit on the United States, there are 84 individuals who are in the United States, and here's where you guys are located. So the darker blue means that there's more of you, and the lighter blue just means that there's less that are participating in Simons Searchlight. So I talked a little bit about collecting blood samples. So we have 31 individuals who have contributed blood samples. Now you'll see that the numbers on the opposite side are a little bit lower. Sometimes that means, and you may have noticed for those of you who've done a blood sample, that we try to draw more than one tube, and sometimes we don't get them all. So if you were at another family meeting, or if you wanted to see if we got sufficient samples for you, we could absolutely look that information up from you. Just get in touch with us to see whether or not you might want to have another sample drawn. And looking at the induced pluripotent stem cells, so there's a handful of variants where we're working on trying to generate induced pluripotent stem cells for some variants in MED13L. I don't know the research group off the top of my head, but there is one research group that has in fact requested to use MED13L induced pluripotent stem cells. These can be requested, the ones that are available. So let's take a look at the medical history data, and then we'll take a look at the behavioral data. So as I mentioned before, all the data I'm presenting today only includes those who have the pathogenic or likely pathogenic variants. Those small number of individuals who have variants of uncertain significance are not included in this data. So in terms of the age, you'll note that most of the individuals that we know who have MED13L are on the younger end. A lot of that has to do with the progression of our ability to do genetic testing over time. So there are very likely individuals who are at the older age range who just never really had access to genetic testing when they were younger because we didn't understand how impactful that could be. So those of you who, though, see yourselves represented at the higher end of this, note how valuable your participation can be to help us better understand long-term outcomes. Because right now, most of the individuals we know who have MED13L are under the age of 10. The kinds of developmental and behavioral concerns that we see. So you'll see that most, if not all, individuals with MED13L have some kind of developmental delay or intellectual disability. There are a portion, but not all, who also have autism spectrum disorder. We also see more diagnoses of anxiety, ADHD, and also OCD. In terms of medical issues, there are a range of concerns. I will just draw your attention to the ones that are closer to the top. So more significant endocrine concerns, gastrointestinal concerns, more surgeries, vision concerns, as well as more neurological concerns, largely represented by hypotonia kinds of issues. And you can see that well represented on this graph that the neurological conditions are almost entirely represented by hypotonia and coordination problems. There are about 15% of individuals who report having seizures. That is to say, a minority, but a substantial minority who will present with seizures of a variety of different types. Vision vision problems are not uncommon as well. In particular, nearsighted and farsightedness, crossing of eyes, and astigmatism. Endocrine concerns, largely related to being a little bit smaller, related to either short stature, and then some individuals who are noted to having failure to thrive. There are some orthopedic concerns, but not remarkably common in this population. And then heart concerns, again, not remarkably common in this population either. We do have some that individuals have occasionally gone on surgery here. So this can include things like PE tube placement, tonsils, and adenoids, which is kind of what falls under that head and neck category. And then we do have a number of individuals who are on medications, which includes largely for sleep, GI. In terms of the seizure medications for that smaller portion that are on seizure medications, there did not seem to be any seizure medication that was noted to be remarkably the most effective. I do think I might be a little bit running out of time as I'm trying to keep my eye on it, so I'm going to try to go a little bit quicker through my last slides here. This is a trajectory of how individuals do with their expressive language development. So overall, what this graph is showing is that there is quite a bit of variability. However, we do so that over time, while progress is a little bit more slower and steadier than we would expect in some other populations, that children do tend to make progress over time, and that most individuals are likely to be using some degree of language by age 4 or older. Now, this only includes verbal language. It does not include our children who are on AAC devices. In terms of personal care skills, again, seeing the same trend that over time, while the progress is more slow and steady, we will see that individuals are making progress over time. The same is true with social development, although this is a little bit of a flatter line, that this progress seems to be a little bit slower than some of the other areas that we looked at. And gross motor development, again, slow and steady, staying behind where their same age peers, but definitely making progress over time, and that almost all children are walking by about age 4. And then the child behavior checklist, which looks at just overall behavioral and emotional concerns. The most common behavioral concern that we see is having specific phobias, having a desire for frequent attention, being anxious, like scaring to attempt new things, and being clingy. Uh, for older individuals, again, demanding a lot of attention, asking perseverative questions, having obsessions or symptoms related to ADHD. So overall, just as a quick summary, overall common issues is intellectual and developmental delay and language delay in nearly everyone. Most common medical issues include vision, like astigmatism and crossed eyes and farsightedness, and then hypotonia and coordination problems. Other issues to keep your eye out for, and that that they're at higher rates, but not as common, include seizures, autism, endocrine, orthopedic issues, heart issues, adaptive skill development delays, specific fears, and demanding frequent attention. So I think I just took it up to the minute. So I will stop there if we have time for questions, but I will defer to see if we do have time. I'm happy to take some if we do.
Um, I'm going to ask the one question in the chat because I think I might know the answer, but I would like confirmation. One of our parents is asking about blood draws for Simons Searchlight and availability outside of the US. And I think that's a no, but I'm going to let you comment on that just on your IRB structure and consent and all that other stuff.
Yeah, so we have been trying. The major issue is that having biospecimens cross international lines has been the issue. So
Yep.
according to our IRB, we are able to do it, but the problem is that then the biospecimens sometimes get stuck in customs,
Yep.
and then we can't get them, or we try to. So it's just we've had some problems with customs is the major issue. If we solve that problem, we will absolutely offer it to international participants. If you come to one of our meetings, it's a non-issue. So if there was a meeting in the United States, we could absolutely do it. It's just with the shipping has been the major barrier.
Yep. And I'll just echo that just from that, right? There's a Med13L's partner with Combined Brain with their own biorepository, same issue for collecting from overseas is it's getting a biospecimen back to the States, and it still be, usable, and have the right integrity to it. So it just presents its own unique challenge. We are hoping the second follow-up question is that Simons will be able to come in July for our family meetup along with Combined Brain, so we can continue our partnership and just getting all of the blood with one stick at one time, to make it easier for
Yep, and
folks.
I'll just to echo that too. We've had a wonderful time working with Combined Brain. We have this really awesome partnership now where if you want to participate in both studies, we do one stick. Sometimes Combined Brain brings their phlebotomist, sometimes we bring ours. We just work it out. It's been really great. And so if you want to participate in the most research and not have your child be stuck multiple times, come into the meeting, I think is a great way to do that.
Yep. So yeah, we'll continue to plan for that for next summer, with Erica and the team over there that helps with that. So, and if you don't, for those of you who don't know, Cora was part of the 2022 speech and cognition team along with Marissa Mitchell. There was a recent publication that came out on the speech aspect of that. And I know Cora's still been working closely on some of that. So hopefully we'll be following up on that as well. Um,
I
I'll
just wanted to thank you,
oh,
Cora. And
oh,
if anyone has any questions, if Cora, if you could just look in the chat for the next few minutes.
I can do that. Thank you.
yep.
Thank you.
Thanks, Katie. That's what I was going to say. And then we'll move on to Caitlin Hudak and some EEG, hopefully some interesting EEG data from 2022.
Yeah, I hope it's interesting too. We, okay, can you see that screen?
Yes, yep, we can.
Okay, awesome. And I'll try to watch the chat, but feel free to just interrupt me too. I'm happy to do so. So I'm also presenting on behalf of Niccolo Pini, who is, I think, on a plane right now on his way to Europe. So he couldn't be here today. He was really bummed by that. So we received funding from Simons to come to the 2022 meetup and do data collection. So both of us are developmental cognitive neuroscientists who are kind of like obsessed with how the brain works. Nico he's actually no longer a postdoc. I need to update this intro slide. He's a research scientist at Columbia, and I'm an associate professor at the University of South Carolina. And I've been working in the rare genetics space for about 10 years now. I think that this is my 10-year anniversary, where I started as a postdoc and have really kind of grown. And so I work with a bunch of different genes, and I was really excited to kind of, and continuing to be excited to be more involved in this group in particular. So what I thought I'd do today, oh yeah, I think actually, let me make sure I move on. Just to kind of show you a little bit about what we kind of focus in. Nico also has a lot of expertise in really, really early development. And we kind of divided and conquered on a lot of this work. And it's been a really fantastic partnership. So hopefully you'll have a chance to hear from him sometime soon. So I'm going to kind of give a fairly brief overview of kind of like some of the things that we're doing and why we're doing them, because we have a lot of moving pieces. So there's a lot of like work that's in progress right now. And we're really kind of coming up to some final data sets that we're just not quite ready for today, but I am happy to kind of share the work that we have been doing. And I don't anticipate the results you'll see are going to change particularly much, but we take an approach in my group where, we kind of sit and think through all of these different pieces of what makes up a person and how we're able to navigate in the world. And so when we think through kind of some of our kind of more smaller biological pieces, like how our genes are encoding different proteins that then create cells that build these circuits and networks and physiology, these are really amazing things to test in preclinical models and kind of basic science labs, because you can do a lot of high throughput screening to see what kind of drugs work and the efficacy of how that changes. But when we think about our kids, a lot of times we're really trying to kind of sort through much more complex pieces that have a clear clinical or medical diagnosis, like seizures or GI issues, or kind of the work that we do that's really like involved in the autism space. But it's harder to connect these clinical diagnoses back to those things like circuits and cells. So we kind of do the best that we can to kind of measure as many different pieces of these across the spectrum that we can to best understand what's going on in our bodies and how that's changing and shifting over time. So my postdoc was with Sarah Jane Webb and Ray Bernier, and we work with Evan Eichler's team. The study is now being run by Emily Newhouse. And so we've been collecting data from very intensive kind of phenotype clinical data from families since early 2010s. And since I've branched out and created my own lab, I've called this now the biogene study. So that's what we were doing in Baltimore. And you can see we've kind of been across the board, but our true aim with the biogene study is to kind of create biomarkers of brain processing that are going to be potentially unique in these different rare genetic groups. So this is just some of the data that I'm going to show later. We actually have a little bit more that I'm not representing here because these are saturated or stratified so that these are genes that we know are disrupting the function of the gene. So you can see we have 13 individuals with MET13L in this group, but we have a lot of different kinds of genes. You can see a lot of SCN2A and GRIN2B from some of my other work. And what we tend to see is that in these groups, we're seeing more intellectual disability and kind of more struggles and kind of daily living skills, which are exactly the data that Cora just presented. So I just keep that in mind. So for those of you who weren't able to be with us, I think that these are actually, I'm sorry, I didn't pull MET13L kids photos for today, but these are some of the kids that came through while we were in Baltimore. And we had this amazing little network set up where we had 2 different rooms and we had families coming and going, and the kids would put on these kind of wet sticky nets. And this is how we were kind of recording ongoing brain activity. So we ended up seeing 11 MET13L kids at this meeting alone. And so you guys were a very impassioned group. It was really fantastic to see everyone. And I think word spread. And so people were coming up, and we did our best to schedule as many people as we can. But I'm also presenting on other cases that we had collected as part of the Tiger study as well. So one of the biomarkers I'm going to share today is going to focus on attention and specifically how attention signals shift. So I wanted to just briefly talk about why we think that that's so important. And maybe at the outset, the most important part is the fact that attention is something that all species have. So this is something that we can test in every single animal and kind of preclinical model. It's something that helps every person kind of navigate the world, is how our brain tells us to orient towards something. And so if I were to give you a picture like this, you might be looking at all these cute little faces, and maybe after a little bit of time, you realize that one of those things is not quite like the other pieces. And what's really kind of cool about our attention system is also is a additional pictures. Now that you kind of know the trick, you're going to be a lot faster at kind of picking up on where the kind of pop out or the new thing is. And so we can track not only attention signals, but we can track how this is changing over time. So yeah, I guess is a little bit redundant, but our 3 primary tasks that we've done are kind of looking at just basic sit and still and be chill, which we call a resting state, which tells us a little bit about just how, when you're relaxed, how is your brain organized? And then we have these 2 kind of auditory attention tasks. And one is more specifically focused on speech, but today I'm going to focus on the other one, which is more general and involves tones. So what does this look like? So kids sit and are wearing this kind of wet, sticky net. It only takes a couple seconds to put the net on and get ready. And then they sit and watch videos. They can look at their iPad. They can play on a puzzle. And we play these different sounds for them. And you can see that some of these sounds are repeated over and over and over again. And these are kind of what we call frequent sounds. But then we have a couple of other categories where it pops out a bit. So we've got tones that are a little bit different. In this case, this is a lower tone. But then we also have kind of novel sounds. And I don't think the audio is super great on this. And so you might not be able to hear, but hopefully you can see that this kiddo is having a great time. And one of the things to note here is too, this is in home. So we also have the capability of not doing this just at the family meetings. In the past, we've driven around the country and collected data in individual homes. So what happens is when you hear a sound, your brain has a specific attention response. And what we track is actually the electricity. And if you kind of focus on this red line, this is like that pop out attention effect. And what we know from some earlier work in autism is that we see this much bigger response in autism, where it has this kind of bigger pop out effect. And I pulled some of the data from our kiddos with MET13L, and it looks a little squigglier because there's fewer kids that are going into these averages. But what you can see is that red and black line are very different. So they're separable right around the time where we're having this kind of attention response. So we know that it's there, but to be honest, it's not as big of a response difference as we see in autism. So this is actually, in terms of like the size of that difference, it's more similar to our non-autistic controls. But what we can do is take each of those kind of peak points. So every time you hear a noise, we look to see, okay, well, here's what happened at that time point, and we can take each of those values and then plot them over time. And what we find is that in our kind of control cases, and maybe it's not very obvious to you, but I can tell you, this falls off quite quickly. So it's likely the case that non-autistic kids realize, okay, these sounds aren't very meaningful, and they shift their attention away to other things. So they become less attentive. There's still differences. So you see how the red and black lines are still different, but it's a much smaller effect. Whereas in autism, we see this kind of like sticky response where you don't have that quick of a fall off. So they kind of are stuck on this information, which can then lead to problems, right? So if you're stuck on information that isn't really meaningful, so for instance, thinking of being in a classroom, then that can really cause some problems with how you're taking in information because you just have too much kind of stuck in your head. So what does this look like in Med13L? Well, first we took this N1 response, which you see, oops, I lose my mouse here. So this is a negative going response. So we'd expect the blue line to be much more negative. And we actually see very little difference between these conditions. And it's pretty flat. And when we look at that other kind of pop out red effect, also, again, very flat. So over the course of the experiment, we're seeing very little change in how kids are responding to these sounds. So to us, this could be a metric of kind of understanding how, as information is coming to us, how are we kind of tracking those pieces? And one of the challenging things that Cora also kind of mentioned is that, and I didn't really emphasize on our kind of overview slide, is that our kids with Med13L were not just kids, right? We had a really wide age group of people, and there's true maturational effects that just plotting this out for you today is not quite picking up. So those are some of the things that we're working on now that I wasn't quite ready to show you. The other thing is that we're looking at is for each person, we can see how big that response is, and we can track how steep perhaps the change over time is. And so that's what we're in the process of kind of mapping these back onto, because it really only matters if this is going to map back onto something that's clinically meaningful. But we still think it's really important to kind of understand these kind of patterns of what we call habituation, because they are this nice marker of learning. And part of the reason why I'm so excited about learning and attention signals is that this is an area where we can really look across that little pyramid again to understand how we might predict and see these different changes in other animal species. So I've been working with Annetta Schenk at Radboud Medical University and her student Laura Block for a couple of years now. This is actually Laura's dissertation, which is just about, it should be as a preprint in the next couple of weeks. They even let me come over to the Netherlands and sex flies. I was not very good at it, but it's been a really exciting partnership to kind of think through how are these attention signals different in both the kiddos and the flies. And I'm not going to oversell flies today because I'm only going to give you a little bit of things, but I do think that even though they are not a vertebrate species, they can be overlooked for that reason. But hopefully this is a beautiful review paper that their group put out. There's a lot of great things we can do. And in particular, we're going to focus today on kind of this habituation response, but also they're truly inexpensive. You can do high throughput drug screening and also look at different kinds of knockouts. So for the purposes today, we're going to kind of focus on like how habituation at this like middle level kind of translates between the two. So what Laura did is she took these fruit fly knockdowns, and after kind of breeding them, she put one fly at a time into these little light tubes, and she would load them all up and then turn the lights out so it was very, very dark in the room, and start a computer program that would flash a light at the flies. And what happens is that when they see this light, it's really aversive and they jump. And she's able to record not only did they jump in response to the light change, but at what point did they stop jumping? Because just like the kids' brain signals, at some point in time, the flies realize, oh, this isn't actually hurting me. And you can see they kind of slow down and stop jumping. So she did this with many flies, and we focus in our paper on the 25, 26 or so that where I had human data. So, and she found in those, there's these 3 different patterns. So first is this atypical kind of response where you see that sticky attention, where the flies are still jumping, even though it's really not a harmful thing, and they should kind of start to change their attention to it. And you can see some of the flies that kind of fit this pattern. We only had one gene group that had the same kind of jumping pattern, both in the knockdown and then the control fly, and that was related to CHD8. But where we saw the Med13L, which is SKD as the fly ortholog, was in the severe phenotype, where they might jump really, really rapidly at the beginning, but very quickly they're not jumping anymore. And so here on the left, you can see it's another fly ortholog that's in that same category. We're calling this kind of a severe habituation phenotype. And so you can kind of see how quickly they kind of jump, and the yellow line is lower than the other line. And you can see it takes fewer trials for them to kind of reach this habituation or kind of non-jumping point. And it's not necessarily that it's just about startle reactivity. It really seems to be about this kind of attention habituation. And so here I pulled out what the SKD fly looks like. And was no difference here in this part front between the two, but looking at the slope across them is where we saw the major difference. So basically, when we kind of come back and try to understand both the SKD fly and the human phenotype, we're seeing this kind of alignment where maybe the reason why I'm not seeing any changes in here is because this is a very rapidly habituating response in both the fly and the human model. So that's what we're kind of trying to start digging through, is kind of understand also then what is this functionally related to. So is this habituation pattern related back to any of the autism phenotypes? What sensory behaviors? And then the age part is going to be a really, really important piece. And I'll kind of allude to some of the work that we're trying to do to address that in a minute here. But just kind of a big picture of like where we're headed. So we have these beautiful models in the fly, and we also have a lot of data from our kids and individuals with Med13L. So the idea is like, okay, so if these are both going to align together where we're seeing similar things, and here we saw our Med13L and that severe phenotype where both had very rapid early kind of habituation versus the sticky attention in this atypical group and that non-affected group. So if that's the case, well, with the flies, we can do all sorts of amazing kind of high throughput screening. Whereas in the kids, we can do things like look to see how social interventions are actually changing and shifting and maybe helping habituation patterns in the kids, and kind of using both of these strategies to kind of hone in on what is the right treatment. Because, it's not important if the flies respond. What we really want to know is what's going to help our individuals navigate the world as best they can. So where we're headed and kind of what's in the middle of happening right now is really starting to kind of validate these markers. As I mentioned, that these papers are about to get kind of put as preprints. But we're also really trying to dig out and understand this role of development as well as puberty. So first, just kind of thinking through that pipeline. This is kind of a schematic of what we did. So the kiddos heard these sounds, and you see like the blue and the red things are a little bit bigger than the repeated sounds. We expect those to be kind of a pop-out effect. But the flies saw the same thing in a very kind of regimented way. So one of the things that we're both working on in our respective labs is to say, okay, well, if I show kids a flashing light in that same pattern, do we see the same attention signals? And they're working on jittering and looking to see if the flies are going to respond to sounds in the same way that our kids do. So to try to understand and really refine the way that we understand these two pipelines. The other piece is that, and I think it's really evident in the flies, this is a very fast response. So with EEG, it's hard to really see that when you're looking across 8 minutes of time. So we're working with some computer scientists and engineers to try to understand the first like 30 seconds of EEG data and better understand these really, really early rapid periods to understand if could we have a common marker in terms of this like trials to criterion, for example. So the other piece is that we have funding for a different gene group. So we're working with CEPP1 Society to collect data for them. But that means that we are starting our road trip show up again. So we've identified a couple of hubs where we know that people are. And the idea is if we're there and working with other families, but there are Med13L families that want to participate, we'd love to see them. So right now we're located in South Carolina, so Georgia, Florida are for sure easy for me and our group. My family's from Ohio, Pennsylvania, so I was excited to see Cora's map and see there's a lot of families up north. So maybe I bring a system with me over the holidays, and we have a couple like pop-up shops to do EEG work. And then we are starting to think through a larger push. This would be our first time in Europe. So we know of some family meetings, and we've been invited to these family meetings of other groups in Amsterdam, Athens, and then they want us to do a road trip around France. So we're kind of working out to think through how we can fund those operations. But I just wanted to let you know that I will keep everyone posted. So I can send up a signal, oh, we're going to be in, Denver this time period. And if there's families nearby that would want to come, they are welcome to come participate. So the other piece, the reason why this is so important, puberty has a huge role in how our brains change. And I really want to try to get to that point of what I think Cora mentioned too. We really want to understand the natural history or how people are developing and how this might look different in Med13L. So these are just kind of a plot. The green little points are just different kids that we saw. And I think that these are the 14 that we've worked with across the different studies. And so we'd love to see people and kind of repeat testing. So we're working on what would need to be a much larger grant to kind of support some of this data collection. And we are working with existing data in Simons Searchlight. So some of the Searchlight team is actually working with me to understand how pubertal development is changing and shifting in Med13L. So we should have some updates about that in the next couple of months as well. We also are kind of moving and thinking beyond attention. Attention is great for kind of this cross-species piece, but and I actually don't know that I've heard too much from the Med13L families about emotion dysregulation, but we know it's very common and hard in intellectual disabilities and other neurodevelopmental disorders. So we're working on thinking through creative ways to measure that. And we have a really cool new paradigm, which I'd be happy to talk more about at another time or offline. But we're also working to characterize both the excitatory neurons and the inhibitory neurons in the brain. And using that still and chill measure, which we called resting state, we can actually look at the relative ratio by which neurons in our brain are working. And so we have these glutamatergic neurons, which are kind of what we consider excitatory, versus GABAergic inhibitory neurons, which are kind of helping damp down. So it's like ramp up and damp down. And like what we want is an ideal balance in the brain. And if we had a person who had more of an excitatory response, we'd tend to see this kind of slope look more like this little dashed line. Whereas if you have more inhibition, we would see the same brain signal kind of at a much steeper rate. So this is a really cool thing that we can do because we can actually measure individual differences in this kind of like seesaw ratio. And why I'm super, super excited about it is because Laura Block has already actually started to do some of this work to look at what if we only knock down the Med13L ortholog in excitatory neurons. So we can actually do this neuronal subtyping in the fly and have a much better sense of these kind of cellular mechanisms that have a really clear kind of map back on to the human brain as well. So long term, like where I see us really kind of like hitting is understanding how these biomarkers are going to truly lead to kind of how we should be tracking clinical trial work. And then I just want to also kind of plug the fact that we are doing some might consider weird and wild stuff in my lab, where we're not just recording one brain at once, but understanding how two people interact. And this is a way for us to really understand kind of more complex social relationships. So understanding what maybe some of the social cognitive differences are in this group, and understanding how your kid reacts to you and how you react to your child, and how that kind of reciprocal process might look. We also see this as a potential avenue to involve siblings and understand that sibling relationship, because we have a lot of evidence that that can be one of the strongest social connections for our kiddos that have these rare genetic conditions. So little teasers of kind of things to come. I have a lot of kind of thoughts about like, what do we really need? Funding is always an issue for us because we have to do this like depth of data collection, and this is an ultra rare group. And it never feels like the right stopping point, right? So like we have 13 kids with MED13L. I mean, I know 13's in the gene name. Should we stop there? But we always would like more to be able to really address both the within person changes of how a person is developing over time, but then understanding how maybe different variants have different effects, and really understanding all of these pieces in that kind of piece. We also see that there could be this opportunity for using EEG in clinical settings, because our system's very expensive and kind of tricky because it's a research EEG, but hospitals do EEGs all the time. So is there a way for us to also use those records, or also work with clinicians to kind of do quick assay EEG and use that with a few channels to kind of really propel what we're doing with some of that excitatory inhibitory work? Okay, I think I also finished right on time, but I'm happy to take questions either in the chat or now if there's time.
Caitlin, I'm just going to ask one
Yep.
one question. You actually already posed the other questions I had, we don't know. But so there seems to be perhaps maybe some hearing impairment, or some other maybe structural issues that are impacting hearing capabilities in the MED13L population that we're trying to figure out and uncover. And with some of these tone studies, like how much do you think that is impacting these results here?
Yeah, so because we are that weird and wild lab that takes our system everywhere, we don't have as much control over environment to environment. So it's something we had to solve very early on, where we really are using the data within a person. So it's not necessarily that, let's say my P3A is really, really high and Cora's is low. Like what we would do is look at the relative ratio. So you're looking at my red to my black line, and you would look at like Cora's red to Cora's black line. I'm sorry, Cora, you just became my model for all of this. But so we're looking at kind of within person differences. So if there were known auditory system issues, it would still be relative, because it's not that you're hearing the pop outs differently. So it's just a metric of how we choose to analyze our data.
Okay, so you basically use yourself as your own control and
Exactly.
looking at that response. So then you can measure the difference between the two and how significant that is. So you're, in a way, already trying to compensate for perhaps a hearing deficit that may not be known.
Yeah, and we could test to see if there are broad differences, but I would be a little cautious of that, because the environments were always a little different, which but if we saw like a really across the board problem, we would be able to tell. across all the
Yeah,
kids.
yes. Yeah. They would all kind of look very similar, right?
Exactly.
Yeah, individuals would look very similar to each other if there was something else going on. So the other thing, I'm just going to ask another question, but I forgot it off the top of my head. So I will have to email you. Oh, I know, puberty. So a couple of things I just want to make a point of there. Um, Anecdotally, the puberty issue is hot in sort of the social media aspects in terms of like early onset in a lot of kids. But when we looked at the Simons data, like there's just not enough individuals. I think it was an N of 10. So there's like essentially nothing there that for me, draws any conclusions on what is actually happening in terms of puberty or changes, and how that impacts, development and EEGs and biomarkers and seizures and all of these other things that are really important. So that's just a plug for like why participation in like the puberty survey really matters, and getting information out there. And then the other comment I wanted to make is actually just on the abstract. I don't know if you were in that, Caitlin, that was published on seizure frequency in the Med13L population, which was it was actually just it was looking at all of the data, all data in Simons in 55 genes that have, seizures identified, and within those that the onset or what the average reporting age was 8. And so, and the seizure presence in the Med13L population was low. It's about 15% and not resistant. And so again, I think that speaks to perhaps if there's something that might be occurring on an EEG, it might be occurring later than what's being documented right now. And so more just plugs for why the research matters, because if puberty is changing this and is impacting Med13L population, that's when we're going to see it. We're going to see it perhaps a little bit later than what we know right now, literature-wise.
And we should circle back on the puberty data because we're looking at that, but we're leveraging all of the like a multi-gene approach and some kind of interesting statistics. So I didn't pull the statistics for this presentation because life, but I think that there's actually I also know the puberty data really well, like how we measure it. And there's some inconsistencies in Simons that I've been talking with the Simons team about. So we can offline maybe chat about that specifically because we might have some insights about that. And I think you're absolutely right. Like understanding that because it's a huge maturational shift and it's also a lifestyle shift. Like our kids are in very different social environments and their bodies are changing and they feel things and they don't always know how to communicate that to us. So so we're a big adolescent heavy lab, even though we are lifespan. So maybe we could have even a whole other webinar where we all just talk about puberty in our different systems.
That sounds uncomfortable. But I think that would be really helpful and maybe help the community also understand a little bit more what they should be looking for and participation and why that matters for things like this because of all of the other shifts that are going to come along with it.
Well, and knowing how to prepare themselves because that's half of it too.
Yes, or 90%. So thank you so much, Caitlin. Feel free to put stuff in the chat or feed them to Katie or I, and we'll continue this conversation. All right, Randy, are you ready to go?
I am.
Excellent. So while Randy's getting his screen up, this is Randy Strick from Rowan University, and then I'll let him explain his project and everything else. You should have sharing capability. Let me know if you don't.
Is it working?
Yeah. Yes. It almost looks like a split screen. There we go. Perfect. Looks great.
All righty. So my name's Randy Strick. We changed our name of our university 3 times in the last 4 years. So now
Bowel movements. Oh, sorry. Thanks, Katie. Um, is not being documented in the literature, but is likely impacting your day to day. And then the hearing you heard, I commented on that a little bit earlier, as did Katie. This is something that you all are seeing in your day to day that is there's 6 cases of any hearing issues at all across 90 plus patients in the literature. So this is clearly something that we think is important for us to evaluate and monitor and watch, and how it is impacting what's going on in and around your child's life. And of course, sleep, which is like never reported for in literature, but it is plays into so much of what happens with behaviors, with seizures, with how social aspects, like everything, just emotional dysregulation, right, of what happens in the day and how those things are connected. So again, just a really high level overview of what you're already contributing and how that is matching up, and where we think the community has a huge value to contribute to the knowledge base of Med13L. I'm just going to keep these for your own learnings in the future. So I'm going to move just to a little bit of science, and then what the foundation has focused on in the last year or two, and then what's sort of on the horizon. And so I wanted to speak, first just to Randy and our team of our scientific advisory board, which we try to talk with them as much as possible about ideas for funding, or where we should go as a foundation and what makes sense. And then Randy, I think I need an updated picture of you.
You think?
Just gonna say there.
Yeah, no. I think that's a little old. You're right. I'll see what I can find.
Thank you. So one of the challenges that Med13L is facing is that this is a really large gene and protein. And what we're finding is that the variants that are occurring across the entire spectrum. And so why that matters is like, and Cora spoke to this too, it contributes to a large variety of symptoms and a huge spectrum of symptoms. And then the blue rectangles here are likely what we would call a hot spot. We are starting to see maybe some coalescing of missense variants in these little prime blue areas, where missense is one amino acid is flopped out for the wrong one. So for the other one. And so those seem to be concentrated in some key areas. And there are a couple publications that talk about some symptoms that might be more present in individuals that have variants in these spots. But those are still being flushed out, and we'll continue to learn more as we get more and more data and information. But I wanted to just share this as like, this is one of the things that we're thinking about and wanting to talk about with scientists and researchers, is how this interplays with how we look to discover therapeutics to help improve, Med13L individuals' quality of life. And Randy talked about this a lot, but this is just a high level overview in a infographic of what Med13L does. I actually made this in conjunction with Alicia Campbell, and like, this is what the normal cell should look like on here on the right. And so, right, what Randy talked about is how intact your mitochondria really should look, right? Should be all together, this, functioning well, nicely, using oxygen efficiently, producing energy efficiently, and then also this gene transcription piece. So making all of these other genes and proteins for other body functions. And what we're seeing is what Randy and Alicia are really uncovering a lot of, is this mitochondrial dysfunction and this gene dysregulation. And so right, Alicia added these scissors, which I think is a very lovely picture of what is happening, that those mitochondria are now being split apart and they're just not working the way that we need them to work for everyday function. So what we have funded and what kind of needs some additional support. So Randy also mentioned Chad Gruder over at Iowa. He has several mouse models going right now, which is really exciting. And then there's a couple of specific mouse models up at Jackson Labs in Maine. They just finished phenotyping a few of those models. So that means they're trying to understand the clinical symptoms in these mice and how they are connected to Med13L, and if they look very similar. So we will be meeting with them very shortly to kind of understand that data, and hopefully then, right, gives us more opportunities on how we can reverse those, right, and make those clinical symptoms either disappear or improve. Lots of other models. We've heard that one of the French researchers have some organoids in process, both for Med13 and 13L. So again, if you remember, actually, I think even Caitlin talked about this. No, Cora talked about this, and Randy. So Med13 and 13L are very related to each other. So they're creating organoids, which are tiny little brains. And again, looking at how those develop. And then we have a zebrafish model also in Poland. Let's see here. Biospecimens, lots all over the place. Simons has a lot of collections for iPSCs. Randy has some fibroblasts that have been generously gathered by the Columbia team, whole blood, plasma for biomarkers, etc. So we're really stocked up here, although we continue to replenish the supply as much as possible, since they do get used, and you can never, well, I think you can never have enough blood on file. Mitochondria research. As Randy mentioned, lots of stuff happening there. We just issued a second grant to try those therapeutics. The antibody validation and some drug repurposing at Caddy King's lab in Vanderbilt. That was a recent grant that just got completed. We talked a little bit about some of those small molecules, and those will be further tested. And then actually Randy's team and then Chad's team are also validating those antibodies, including in mice, just to make sure that we can adequately use these. These are important for measuring Med13L protein. Proteomics. So this is complete. So we had just over 20 families, controls and affected siblings and affected children donate to this study last year. This is done in conjunction with SomaLogic and Combined Brain. This data is being analyzed across neurodevelopmental disorders. So we should have a really nice sort of data set and pieces of information that are biomarkers. So what we can measure and how we can measure change, not just for Med13L, but perhaps how we can identify opportunities to collaborate with other neurodevelopmental groups where we might be able to find some similar therapeutics for indirect pathways. I have to throw this in here because this is a big deal in the ICD, in the neurodevelopmental deal, or in the neurodevelopmental space and genetic space in general. If you have not informed your physician, at least here in the US, of the ICD-10 code, Q87.85, this will help us gather really important data, improving the patient population numbers, where we're located, et cetera. It does have to mature over time because it has to get into the systems and be diagnosed. And then in Europe and other countries, we wait for the WHO to update their codes, and then that'll be available overseas. That should be happening early 25. The next few slides are really just publications that the foundation has been involved in. So this is one I did for Boston Children's and TNC on that strategic research plan and sort of what's been happening on the foundational level. Combined Brain just did a poster for NORD, which we had one of our community members attend. This is on disease concept models, which Med13 is actually in the middle of completing. So this will really provide us great insight to not just symptoms, but caregiver burden, individual impacts, and how that impacts the daily lives of you all and the community. And then Jen Bain just published this at a cerebral palsy meeting, and using some Med13L data where she's has seen many of your kids and had also done some clinical intake along with getting those fibroblasts. So this is just important because the more Med13L is out there, the more it matters. The more eyes are on it, the more interest that there is, the more people that it can help bring into the research and clinical fold to help propel a lot of this information gathering forward. So I think I can get through the rest of this here. So on the horizon, so I just wanted to kind of provide a little bit of a sneak peek to some of the opportunities or thoughts for the next year or so that are all kind of in the works or part of these ongoing conversations that are happening behind the scenes. So drug repurposing. So we know Rarebase got or Transcripta Bio got a hit with naproxen. We're going to do further sort of testing on that. As we talked about, Randy mentioned what he's doing, and then Caddy will continue to test on her drug. And then a new opportunity is perhaps with Unravel Bio, which takes nasal swabs and maps the transcriptome. This is kind of all in the process of, so this is also a high throughput screen, which Caitlin mentioned, but it's basically taking a lot of different drugs and seeing if they will rescue or shift, a clinical symptom or a biomarker or something of that line. So this is something we're sort of talking about and figuring out contracts, either with other organizations, like Med13L or with Combined Brain as a huge group project. Gene-based therapeutics, this is very early on, conversations around potentials for ASOs, CRISPR-A and others. All of these are really focusing on haploinsufficiency. So really looking at upregulating that the one Med13L gene that is still functioning. And so again, this is very early on in sort of what we're looking at in contracts and costs, but this is something that we're focusing on. Functional characterization of neurons. So we don't really have a good idea of what is happening. Actually, Caitlin might be doing this faster than we can do it. So this was it was good to have her on this call. But this will really help us look at those inhibitory and excitatory neurons. So really looking at what is going on there, how those neurons develop, what the impact of a certain variants could do to those neurons and how they're growing and developing over time. So we're still exploring the landing place for this particular aspect, but this will really shed a whole lot of light on some of the brain functionality that we could be seeing. And then big sort of community update. So family researcher meetup for July 10th through 12th in 2025. It's going to be in Boston. We're really hoping that if we have a good East Coast location, we can help encourage some of our families and researchers that we have, especially in Europe, to come and participate in family and research activities. We're actually partnering with CTNB1 and NB1 Connect and Cure. CTNNB1 is a protein that's made through the Wnt signaling pathway, which we know is heavily involved. Med13L is heavily involved in regulating. And so we thought this would be a really nice opportunity to perhaps bring our communities together. And hopefully help understand each other a little bit more and maybe some additional opportunities to collaborate. So hopefully Simons, Cora, Caitlin, Randy, put it on your calendar because you're getting invitations from us anyways. We're excited about that. These are the conversations we're also having behind the scenes for improving care and clinical trial readiness, either through centers of excellence or through a formal natural history study. So we've got a lot of talking going on, and just trying to figure out what makes sense, what sort of our right sort of collection. If you remember the map from Simons of where we have our communities located, that might make it easier to get to one of these institutions. So it's just a lot of conversations and ironing out details, but we've got a lot of people that are interested and we want to continue to facilitate those relationships to really help with care and research moving forward. We've already talked about both of these and why they're so important, and they're complementary to each other. And certainly appreciate the burden that it takes to enter the information on these surveys. But it really does help form decisions around not just clinical care, but understanding the disorder, the trajectory of the disorder, and where we might be able to have therapeutic interventions for the future. This has happened, I think, in the last 6 months. We've made some really great connections with our communities in Spain, with Australia, and Germany and Italy. We've always had a good partnership with our French community, but really having some new families step up from these other countries and lead some efforts there, I think will provide a much stronger community base and foundation for how we can kind of spread some of the opportunities across the world. And I think Caitlin already threw some countries up there on her map. So hopefully, I know we have some of our communities overseas on this call. So we will continue to have those conversations with Caitlin and her team on those available opportunities to gather that important information from you all. And then another thing I really want to focus on from my perspective and my role is really to grow that network of scientists and clinicians and, provide opportunities for people like Caitlin and Randy and Cora to, provide their insight and their expertise, but also learn from others. And it doesn't hurt to have more eyes on Med13L and disorders. And so I think this provides a lot of opportunities to really open the door for that collaboration and knowledge base expansion. And that's it. That's all I have. So really, I was trying to keep it short and sweet and more informational. I'll pause there for any other questions. For any of our researchers, we have a few minutes before we're finished. All right, no questions. We nailed it, guys. Nice job. I just want to say a huge thanks. Oh, is there anything hold on. I just got an alert here. Oh, just some thank yous. I just want to say huge thanks to Cora and Caitlin, who made some time out of their day to talk about Simons Searchlight data and Med13L specific information, and also Randy, who was gracious enough to let Alicia go on a honeymoon and and do the work for her. And coming to present on the work that they're doing in that lab. And as always, if you need anything, hopefully you know where to find us on Facebook, info@med13l.org, and we'll be available. Well, sometimes I'm not very good at email sometimes, but I try. Stop.
Thanks for having us, Rachel, and everyone. Nice to meet everyone.
Yeah, thank you so much, guys.
Thank you.
Thanks.
Thank you very much.
Thank you.
Thank you.
Katie, this says it's paused. Oh, stop.
Vanessa vlog.